ค่ะขอเสียงปรบมือต้อนรับ Professor Roger Kornberg ด้วยค่ะสวัสดีค่ะ Good afternoon, ladies and gentlemen. Welcome all delegates to the special lecture, The End of Disease, by 2006 Nobel Laureate in Chemistry, Professor Roger Kornberg. My name is Sompit Samipak. On behalf of the organizers, we are honored to be the host of this remarkable event. So, ladies and gentlemen, now our distinguished guests have arrived. We will begin the lecture in a few minutes.
เรียนท่านรองอธิการบดีมหาวิทยาลัยเกษตรศาสตร์ท่านคณะวิทยาศาสตร์ท่านผู้บริหารและแขกผู้มีเกียรติทุกท่านค่ะขณะนี้ได้เวลาสมควรแล้วขอเรียนเชิญท่านคณบดีคณะวิทยาศาสตร์รองศาสตราจารย์ดร.อภิสิทธิ์สงสารเสนขึ้นบนเวทีเพื่อกล่าวรายงานค่ะขอเรียนเชิญค่ะ
Ladies and gentlemen, now I would like to invite the Dean of Faculty of Science, Associate Professor Dr. Apisit Songsak-sen, to address the report. Please welcome the Dean of Faculty of Science, Kasetsart University, to the main podium.
Distinguished guests, ladies and gentlemen, on behalf of the Nobel Laureate Lecture Organizing Committee at Kasetsart University, I would like to report on the preparations for the special lecture by Professor Roger D. Kornberg, Nobel Laureate for Chemistry. The main goal of this event is to provide attendees with the opportunity to meet and exchange ideas on the research with Nobel Laureate. In addition, to bring participants to apply the speaker's suggestion and experience as good practice in their work to be successful in their profession. The organizing committee has coordinated and contacted various agencies to organize this event. We have been preparing for the event for approximately half a year by publicizing and inviting participants who are almost 754 people, consisting of 155 participants from higher education institutions, 270 undergrad students, and 149 graduate students. Moreover, we have 148 students, 17 teachers, and staff from our school networks. There are 15 participants from government agency and private sector. We thank all attendees for their interest and time spent at this event. This event aims to provide a unique opportunity for our academic community to interact closely with Professor Kornberg's breakthrough idea. It is an invaluable opportunity to exchange ideas and gain knowledge that will benefit our academic and research pursuit. Under the topics of the end of disease, the remarkable developments in biomedicine and their impact on humanity, our commitment to fostering scientific study for the benefit of humanity. We expect all participants will gain insight from attending the Nobel Laureate Special Lecture and be encouraged to produce high-quality research. We thank our event sponsor, primarily Kasetsart University, for the financial support along with Eurofin Food Tasting Thailand. We would also like to thank the following sponsor, BMW Group Thailand, Kasikorn Bank Public Company Limited, Dusit Thani Public Company Limited, and the Mitsui Fudosan Company Limited. My sincere gratitude extends to all involved in bringing this moment success. On this occasion, I am honored to invite Associate Professor Dr. Ladawan Kuangchit, the Vice President of Kasetsart University, for delivering the opening remarks for this special lecture project.
Ladies and gentlemen, now it's time to listen to the opening remark by the Vice President of Kasetsart University, Associate Professor Ladawan Kuangchit.
Professor Roger Kornberg. Ladies and gentlemen, distinguished guests, on behalf of Kasetsart University, I'm pleased to welcome the attendees to the special lecture by Professor Roger D. Kornberg, Nobel Laureate in Chemistry. This event presents the famous Nobel Laureate scientist for his remarkable contribution to chemistry as part of Kasetsart University 80th anniversary celebration. Today, we are honored outstanding minds and their extraordinary achievements, recognizing the transformational power of knowledge and creativity. I'm honored to coordinate this event with our partners, the International Peace Foundation, to highlight the massive impact of Nobel laureates across various fields. They are the luminaries who have improved our knowledge. Being one of the higher educational institutes in Thailand, the recreation of knowledge naturally motivates and pushes us forward. We now encounter the prospect of the end of the disease. The extraordinary developments in biomedicine and their implications for humanity, guided by Professor Kornberg's expertise. This endeavor resounds with the Faculty of Science, dedicated commitment to science for better life and future. To our distinguished laureate, I appreciate your dedication, which inspires future generations and sets standards for greatness. My gratitude extends to the organizers, sponsors, the International Peace Foundation, and those involved in this event. I thank all participants for attending on this occasion. May this event illuminate the spirit of scientists in both current and future generations, inspiring them to push boundaries and pursue knowledge without limits. I'm confident this event will successfully achieve its objective, laying basis for impactful academic and research collaboration to disseminate the reputation of our knowledge of the land to be widely known internationally. Ladies and gentlemen, I now declare the special lecture by Professor Roger D. Kornberg, Nobel Laureate in Chemistry, open. Thank you.
Thank you very much. Associate Professor Ladawan Kuangchit, thank you.
ค่ะในลำดับต่อไปนะคะขอเรียนเชิญมิสเตอร์อูเว มอรวิทซ์ประธาน International Peace Foundation ขึ้นบนเวทีเพื่อกล่าวต้อนรับค่ะ
Ladies and gentlemen, now I would like to invite the founding chairman of the International Peace Foundation, Mr. Uwe Morawetz, to give a welcome speech. Please welcome Mr. Morawetz to the podium on the stage.
Sawadee Krap, and welcome to the JAPAN-ASEAN BRIDGES event series facilitated by the Vienna-based International Peace Foundation. The events are hosted in cooperation with various local partners, including some of the country's major universities, and I would like to thank Kasetsart University for hosting our event today. Commemorating the 50th anniversary of official relations between Japan and the ASEAN region, Bridges will be continuously held in Thailand and Japan until March, involving the participation of Nobel Laureates for physics, chemistry, medicine, and economics. The Japan-ASEAN Bridges series follows the series of over 800 Bridges events, which the International Peace Foundation has facilitated since 2003 to support education in the Asian region. As peace cannot be achieved instantly, but is a process which needs time, Bridges has not been organized as a single conference, but as an ongoing series of events of now over 2 decades, in which Nobel laureates and international decision makers have built strong bridges with leaders in all parts of society and with the general public. With the basis for peace being education and synergies being the fruit of cooperation, the International Peace Foundation has not realized bridges alone, but has carried out the program together with UNESCO and with 179 other national and international institutions, including 106 major universities and schools across the Asian region. The multidisciplinary and pluralistic approach of bridges, the Philippines, Malaysia, Cambodia, Vietnam, Singapore, Indonesia, Laos, and of the events now in Thailand reflects that peace involves all parts of society. It involves awareness and social responsibility of politicians, the business community, scientists, artists, and the media. And since peace within ourselves, our families, and our environment starts in our minds and hearts, it involves every one of us. In this sense, Bridges challenges us to cross borders and to build bridges by listening and opening up to other viewpoints, by generating new thoughts, by developing innovative forms of cooperation, and by fulfilling the desire of everyone to get to know and to learn from each other. This can lead us to a world in which we will be able to understand each other and the complexities we face together in a new light. So let us be inspired by the knowledge and the wisdom that Bridges continues to offer, an opportunity to get a more inclusive, interconnected, and comprehensive view of ourselves and the world in which we live in, and which we are able to create anew constantly, through dialogues towards a culture of peace which need the participation of everyone. I thank the 2006 Nobel Laureate for Chemistry, Professor Roger Kornberg, who has again agreed to come to Thailand to support the events, and we now look forward to his keynote speech and to his important contribution to build bridges. ขอบคุณครับ
Thank you very much, Professor Mr. Morawetz. So now it comes the time that we've been waiting for. The lecture titled The End of Disease. We would like to proceed by a short CV of Professor Roger Kornberg.
So Professor Roger David Kornberg is an American biochemist who was solely awarded the Nobel Prize in Chemistry in 2006 for his studies of the molecular basis of eukaryotic transcription. He received his Bachelor of Science in Chemistry from Harvard University and his doctoral degree in chemical physics from Stanford University. After his PhD, he did a postdoctoral fellowship at MRC Laboratory in Cambridge. Then he became a faculty member in the Department of Biological Chemistry, Harvard Medical School. At present, he is a faculty member in the Department of Structural Biology at Stanford University.
Thank you very much. That was such a strong CV. So now it is the time for the special lecture, The End of Disease. Ladies and gentlemen, please join me in welcoming the 2006 Nobel Laureate in Chemistry, Professor Roger Kornberg.
So let me begin by thanking most warmly the leadership of the university for this invitation, and Mr. Uwe Morawetz of the International Peace Foundation for making it possible. Thank you very much, and look forward both to speaking to you this afternoon and also to a discussion that will follow. I'm most eager to interact with the members of the audience, especially the young people in the audience. Some years ago, I heard a wonderful lecture by Sir Paul Nurse, who won the Nobel Prize in Medicine or Physiology for his studies of the eukaryotic cell cycle. The title of his lecture was Great Ideas in Biology. That may sound like an oxymoron, which means a contradiction in terms, like jumbo shrimp or military intelligence. We think of physics as the science of ideas, and of biology as an empirical science. Nevertheless, as Professor Nurse explained, there have been great ideas in biology. And I will mention 3 by way of introduction to the rest of my lecture. After that, I will speak briefly about my own work concerning the production of messenger RNA, which many of you know from its recent use in vaccines against virus such as SARS-CoV-2. Toward the end of my lecture, I will mention a practical application, again with the use of RNA, but this time not for its production, but rather the use of RNA to achieve its own destruction. The first of the great ideas in biology that was mentioned by Professor Nurse was the idea of the cell. So if you could, the first slide for me, please. Well, while I'm waiting for that slide to come up and show you, there we go. The first of the great ideas, the cell. The cell we can attribute, so its discovery due to Anton van Leeuwen. The cell is the unit of life. There are 100 trillion cells in a human being. There are 200 different types of cell in a human being. An important point that I will come to later in this lecture. Leeuwenhoek was a tradesman of no special education in 17th century Holland. He became interested in grinding lenses in order to achieve magnification. And with the use of those lenses, in ordinary material, he discovered the existence of microbes, of cells from human beings, such as blood cells, sperm cells, skin cells. Leeuwenhoek was responsible for the development of microbiology. He was the founder of the subject of microbiology. The second of the great ideas in biology, due to the lecture from Professor Nurse, the idea of genes. They were discovered by an Augustinian monk, as many of you will know, Gregor Mendel, now more than 100 years after the time of Leeuwenhoek. Mendel would walk in the monastery garden and observe that there were multiple varieties of ornamental plants. And he asked whether, if planted next to each other, whether the characteristics, the traits of one would be passed to another. He discovered that does not occur. he found that the plants breed true, that the offspring of a plant are identical to their parents. He went on then, found the subject of genetics to discover the existence of heredity, and finally to discover how traits can be transmitted from parents to offspring in certain numerical ratios. He discovered the laws of heredity. He published his work toward the end of his life, but it went unnoticed, and it wasn't until the beginning of the 20th century that it was rediscovered and its importance appreciated. The last of the great ideas in biology that I will mention that serve by way of introduction to this lecture, the idea of life as chemistry, the idea that all of life can be understood in chemical terms. This we may attribute to a great French chemist, Pasteur, also during the period when Mendel did his work late in the 19th century. Pasteur, along with Robert Koch, is credited with the idea of the germ theory of disease, a very important concept which led, amongst many things, to the introduction of sterile surgery by Lister, and in the case of Pasteur, understanding, as he did, that bacteria cause the spoilage of beverages. He was a man of great renown, and when a problem arose in the wine industry in France, his advice was sought, and he discovered that wine is produced by the action of microbes, in this case, yeast. Yeast are responsible for fermentation, the process whereby sugar is converted to alcohol in the production of wine. Pasteur then asked the great question of his time: what is life? What is it that distinguishes a living thing from an unliving and inanimate object? We all know there is a difference. We immediately recognize whether something is alive, and we instinctively know what it means, the opposite, a piece of stone or a boiled egg, obviously inanimate. Pasteur asked the question, is life something indissociable, something that cannot be separated from the living organism itself? Or is there a vital force that resides within the living thing, which could be identified, could be isolated, could be separated, shown to be responsible for life? Pasteur then performed one of his many great experiments. He broke open the yeast cell and took the contents, of course, not obviously alive, asked, could they cause fermentation? Could the inside of a yeast cell convert sugar to alcohol? Pasteur got the wrong result. Pasteur got the wrong result. No fermentation occurred in his experiment, and he concluded that life is something associated with the living, breathing thing itself. So great was his influence, no one dared to repeat or question his work until a quarter century later, when Edward Buchner repeated the experiment and got the opposite result, which was the right result. Buchner found that the contents of a yeast cell are capable of fermentation. We now understand Pasteur failed because one of the many enzymes responsible for fermentation was damaged during isolation in his experiment. Buchner used a different strain, a different species of yeast in which that enzyme, that protein catalyst, responsible for the conversion of sugar to glucose, survived the process of breaking open the cell and recovering its contents. And for that important discovery, Buchner won the Nobel Prize in Medicine or Physiology in 1905. Buchner discovered that enzymes, protein catalysts, small protein molecules that facilitate the reaction of fermentation, the conversion of sugar to alcohol, represent the vital force. Indeed, every activity, every action, every capability of every living thing is dependent entirely upon the action of enzymes. I will speak to you now about my own study of one of the most remarkable, arguably most important of all enzymes, the enzyme that reads genetic information. So I will speak to you about an example of life as chemistry, in this case, genetic chemistry. So an enzyme that acts upon the genes discovered by Mendel to derive the information that resides within them to direct the formation and the function of every living thing. Now, most of you know that genes are made of DNA, and you are probably almost all aware of one simple essential fact necessary for the rest of what I will tell you, which is DNA is so-called double helix made of two strands that are wound in helical fashion around one another. Along the length of each strand are building blocks, 4 types, and it is the order, the sequence of those building blocks of DNA that encode genetic information. The enzyme that I have studied is responsible for the process of transcription, whereby the information in DNA is copied into another very similar molecule called RNA, which carries the genetic message for the ultimate production of the proteins, the enzymes, which I have told you are the basis of life. The enzyme RNA polymerase, so called because RNA is a long polymer, copies the information in one strand of the DNA double helix I understand that I perhaps couldn't be heard well in the back, and I apologize. I could start over, but in the interests of time, I'll continue and hope to do better. So, RNA polymerase, the master enzyme of gene expression, synthesizes, it generates, it creates a molecule shown here in blue of RNA, copying the information from a strand of the DNA double helix into another long polymer of a similar type. It is then the information in that polymer, the RNA, the blue strand, that directs the synthesis of proteins. Proteins are also long molecules, long linear molecules, except in this case, the components differ from those of DNA and RNA. They are building blocks called amino acids, but the protein has the capacity to coil up in a compact form which endows it with its catalytic capability. In the case of fermentation, its capacity to rapidly convert sugar to alcohol. Now, in the case of RNA polymerase, a very large protein, it is composed of nearly 30,000 individual atoms. We set out some 30 years ago to discover the arrangement in space, the three-dimensional structure of RNA polymerase, the configuration of those 30,000 atoms. This we accomplished by the formation of crystals and then irradiation with X-rays using the facility called the Stanford Synchrotron Radiation Laboratory, where a 1 mile long linear electron accelerator gives rise to X-rays intense enough to reveal the arrangement of atoms in RNA polymerase. With the result that is shown here, not only the pattern of the long protein chains in white and gold, but the RNA polymerase in the act of reading genetic information. In this image, you see the DNA double helix, so the gene entering from the right, unwinding, as you recall in that schematic, to allow one strand, that in blue, to serve as template for formation of RNA in red, assembled one building block at a time at the so-called active center in the middle of the RNA polymerase molecule. RNA polymerase is the master enzyme for gene expression, for the reading of genetic information, but it is a dumb molecule. That is all it can do. It is capable of reading information with extraordinary accuracy and efficiency, but without distinguishing one gene from another. You may recall my mention early on, we as humans are composed of 200 different types of cell. But you may not know, every cell in the body contains the complete genetic information. Every cell contains DNA sufficient to instruct the formation of the entire being. So what is it that distinguishes one type of cell from another? And the answer is, it is selective reading of the genetic information, reading only the genes in muscle needed for its formation, but not, for example, the genes for nerve or for skin or for blood in muscle. And conversely, in other tissues, only the genes needed in those tissues. We and others sought for decades to understand how this selective reading of genetic information occurs. It is important because it is the basis for human development, and it is mistakes that are made in reading, in the selective reading of information, that give rise to diseases such as cancer. What we discovered over the years, my students, my colleagues and I, RNA polymerase is assisted by many additional proteins in the process of transcription. So the RNA polymerase itself is made of 12 individual proteins with a mass of 500,000 mass units. There are, in addition, so-called general factors, another 25 proteins. They add another 1.5 million units of mass. And finally, what we call mediator, that I will explain in more detail in a moment, that is responsible for the selective reading of genetic information, another 20 proteins, another million molecular weight of mass, nearly 60 proteins assemble on every gene prior to every event of transcription to achieve the appropriate expression of genetic information, the production of the correct RNA and protein at the right place in the body at the right time and the amount required for function. I'll just briefly mention an example of a general factor and then one mention further of mediator. One of the general factors we discovered performs another different essential function. So you may not know that your DNA, my DNA, everyone's DNA is subject to damage at an extraordinary frequency every day. 2 quintillion events of damage to your DNA and my DNA every day. That is to say, 2,000 billion million events of damage every day caused by radiation, sunlight, food additives, reactive oxygen from the atmosphere. 2 quintillion DNA lesions in every person every day. Only 2 lesions are required to produce cancer. So all of this damage must be repaired every day, DNA, every single day. One of the general factors that we discovered required for reading genetic information also repairs damage to the genetic information. This particular general factor, a repair factor, ensures that almost none of those DNA damage events ever persists in our genome of any of our cells. I said I would also mention just add one brief comment about mediator, which is so important. So mediator, we discovered, solves the problem, answers the question for development, and when it fails, the development of the occurrence of a disease like cancer. Mediator receives signals from inside a cell, from elsewhere in the body, and from the environment. It processes that information and then delivers an instruction to the RNA polymerase what gene to read, as I have said, at what time, at what place of the body, and in what amount, for the purpose of formation and maintenance of our system. And just to emphasize the point about its importance, here's a schematic picture of many different proteins that compose mediator, and you don't need to be able to read, but just see that here a mutations in many of these components of mediator are responsible for many cancers. In this particular example of one subunit, mutations in this particular component of mediator have been traced to bladder, breast, colorectal, gastric, hepatocellular, lung, ovarian, and other cancers. That emphasizes both the importance of mediator and its role as a target basis for both disease, cancer in particular, and ultimately for cancer therapy. Now, I would like, after this very brief account of my own studies, to emphasize to you that the transcription machinery that we elucidated, that we discovered and clarified, that I have just described, was all found in studies in yeast, in baker's yeast, in Saccharomyces cerevisiae, a simple unicellular, single-cell eukaryote organism. That is, everything we discovered in yeast is directly applicable, is exactly the same in humans. For example, the RNA polymerase is identical in its structure that I showed you and in its mechanism, and almost exactly the same in its sequence of components between yeast and human cells. This is not an isolated example. Antibiotics were discovered in bacteria. Genetic engineering discovered in bacteria. More recently, CRISPR-Cas responsible for the possibility of gene editing discovered in bacteria. And an example I will come to later, what is called small interfering RNA, also discovered not in humans, not even in higher organisms, but in a very simple creature, the nematode worm. What I wish to emphasize, all of these discoveries, that of antibiotics, genetic engineering, CRISPR-Cas, and so forth, formed the basis of modern medicine. These discoveries were made not by seeking to solve problems in disease. They were discovered in the pursuit of knowledge for its own sake. They were discovered to satisfy the curiosity of investigators who wished to understand nature. And yet, it was those discoveries made by chance in the course, as I say, of the pursuit of knowledge that formed the basis of modern medicine. So the lesson from the past century that gave rise to these discoveries is, in a way, counterintuitive, the opposite of what you might think. It tells us that to solve difficult problems, to solve great problems, don't try and study them directly, but rather pursue a curiosity about nature, and eventually the solutions to those problems will emerge. Do basic science, and the solution to all of the problems that we confront will eventually emerge. Now, I thought I would take a few minutes to comment especially about two examples: antibiotics on the one hand, small interfering RNA on the other. Antibiotics are an example of small molecule drugs. You all know about penicillin, the best known antibiotic. One example of a small molecule drug. These are molecules of a few hundred mass units. They're so small that they can enter our cells and achieve their therapeutic effect. Small interfering RNA is not so small, as I will explain to you, and it does not readily penetrate cells. And despite its great potential for therapeutic use, it could never be used since it was discovered 30 years ago until very recently, because of an additional important finding that I will mention in the conclusion of this lecture. Now, in the interest of time, I will not speak in greater detail about recent developments in the discovery of new small molecule drugs, but I will concentrate instead on macromolecule drugs, that is to say, DNA drugs, RNA drugs, and protein drugs. These molecules, DNA, RNA, protein, could not be used as drugs until now. You know that messenger RNA could only be used as for the purpose of vaccination because of the discovery made only very recently of how to introduce it into human cells. It would not ordinarily penetrate cells, ordinarily would be of no use, would deliver no therapeutic benefit. The advantage of macromolecule drugs like DNA, RNA, protein, they are informational macromolecules. They are programmable, unlike small molecules like antibiotics, so they can be devised readily for any purpose, and they are completely specific. Small molecules often, in fact, most of the time, small molecule drugs have off-target side effects which can be harmful and limit their use. DNA, RNA, protein are natural molecules. They are evolved by nature to achieve absolute specificity and perform only the desired function. So if DNA, RNA, protein can be used as drugs, they will achieve the ultimate in disease therapy. Now, in the case of, I'm going to skip over these because there's not time, and I'm going to talk to you about small interfering RNA and a particular example of its application, and that is to future pandemic preparedness. This will illustrate both the power of an RNA drug and also a very important application for all of us for the very near future. So I mentioned earlier that our work, studies that I performed over many years with my colleagues at Stanford, were concerned with the production of RNA by RNA polymerase. And I told you that I would comment at the end about the opposite, namely the destruction of RNA, and that is the role of small interfering RNA. Now, future pandemic preparedness is something we all need to worry about. And the reason is that flu, for example, has already caused 4 pandemics just during the last century. And we can be sure that any day now there will be another flu virus that is a pandemic variant that will emerge. In fact, there's great concern at this moment about H5N1 bird flu, which when it has infected humans has caused a 56% fatality rate. Very great concern about an outbreak of this bird flu in a couple of parts of the world. There will be another influenza pandemic unless something is done to prevent it. Now, the solutions, that is to say, the way of addressing a future pandemic, up until what I will tell you shortly, fall in 2 categories. Conventional drugs. These small molecules, like new antibiotics, take many years to develop. RNA vaccines also require time, at a minimum months, to develop for a new virus. Too long to prevent a pandemic virus from spreading rapidly around the world. So we can make a new RNA vaccine when the next pandemic virus assuredly emerges, but by the time we have a vaccine for it, it will be too late because it will already be widespread. Of course, another problem with RNA vaccines that you all know, the challenge of introducing the RNA into human cells for therapeutic purpose is such that cold storage of the special form that is used is necessary. Vaccines are problematic for this and other reasons, as I'll mention. Now I'll tell you about a genuine solution to the problem that will protect all of us from the next pandemic. And it is the use of small interfering RNA. This is a natural method of addressing viral infection. It is more natural than introducing an antibiotic or a small or a messenger RNA or what have you. And that is because the mechanism by which small interfering RNA exerts its effect is present in every cell of our bodies. An siRNA causes the destruction of another RNA molecule that has the same genetic code. So when a new virus appears, in a matter of minutes, we can now determine the genetic code of that virus, the sequence of its genome, in no greater amount of time, using an automatic machine, we can make a small interfering RNA with the same genetic code. And if that is introduced into our cells, it will bring about destruction of the viral messenger RNA and stop the infection. Now, the problem, as I mentioned, is how macromolecules can be introduced in our cells. So a small molecule drug like penicillin, like very many other drugs in use today that you can swallow in the form of a pill, that are orally bioavailable, and on the order of a few hundred molecular weight units. Even a small interfering RNA, the smallest of all macromolecular drugs, is 50 times larger, and moreover bears many electrical charges that prevent its penetration into our cells. We have discovered just over the last short period of time a solution to that problem. So the small interfering RNA cannot cross the wall, the so-called cell membrane that surrounds every living cell. We found that if we attach a derivative, a molecule closely related to cholesterol, to the two ends, it passes freely into the cell. It passes through the wall, the barrier that surrounds every living cell. The way in which this cholesterol derivative is attached results in its rapid release upon entry, and the small interfering RNA is captured as trapped inside the cell. And here is an example to show how well it works. So in this case, a small interfering RNA is labeled with a red dye. If that small interfering RNA did not have attached to it the cholesterol derivative, it failed to penetrate cells. But if derivatized, if with two molecules of the cholesterol derivative attached, then it penetrated rapidly into every cell in the experiment. Finally, I'll show you an example where a small interfering RNA directed against the SARS-CoV-2 virus was used to treat monkeys that were infected with the virus. And the result is dramatic, revealed by nasal swabs. So in the untreated monkey, the dark blue bars, there was no change in the number of copies of the virus over a course of 6 days. But in the monkeys that were treated with small interfering RNA, 99.9% of the virus was eliminated within 6 days. Now that is to be contrasted with the current only drug available for SARS-CoV-2, Paxlovid, which will only result in a tenfold or reduction, or 90% of the virus eliminated. 10% remains. If the drug is removed, you become sick again with the virus. A 1 order of magnitude reduction in viral load by Paxlovid is insufficient to cure the disease. But a 3 order of magnitude, a 1,000-fold reduction in the viral load by an siRNA drug against SARS-CoV-2 is truly curative. It is not only curative, but it is preventative. In an experiment I won't take time to show you, if the drug is first introduced into the monkey, and then they are exposed to the virus, they do not contract the disease. So the advantages of the antiviral siRNA are, as shown here, prophylactic, preventing disease, as well as therapeutic, curing the disease. But what is also very important, this drug, the antiviral siRNA drug, is stable at room temperature. So it doesn't require ultra-cold. It can be stored indefinitely at room temperature. And it is delivered not by injection, but by inhalation. So it is not a vaccine, and it differs from a vaccine in important ways. First of all, it can be stored indefinitely, and it does require a medical professional, a nurse or a doctor, to inject or deliver. Unlike a vaccine, which is at best able to protect 80 to 90% of those who are treated, the siRNA is 100% effective. It will protect every individual. Unlike a vaccine, which takes some weeks for immunity to develop, treatment with the siRNA drug is instantaneous. And the moment it is inhaled, the patient is fully protected from the virus. It is, of course, important in many parts of the world. People are afraid or sometimes reluctant to receive a vaccine. But this is not a vaccine. And so it's likely that it will be widely accepted. And finally, I might point out that, as I said a moment ago, but I should repeat, it can be produced immediately. The moment a new viral strain emerges and its sequence is determined, there are machines for automatic synthesis of the small interfering RNA, so it can be produced and distributed widely. The cost is minimal. A dose of the small interfering RNA will cost only $1. So there's nothing to prevent its wide distribution to protect the population. This is the solution to future pandemic preparedness. It is the way in which we will all be assured there will never be another pandemic. But that is not all. There are 23 common human respiratory viruses. You know of some of them. You know of influenza. You know of respiratory syncytial virus. You know of parainfluenza. There are 23 such viruses, and it is a trivial matter to do exactly the same thing at the same low cost for every respiratory virus. Most of the patients who go into a medical clinic today complain of respiratory symptoms. The suffering of people ill with respiratory virus, not to mention the economic cost of respiratory illness, is very great, and it can be eliminated. Only this method is widely employed. So I've summarized here virtually everything I've told you about the advantages of small RNAs compared with RNA vaccines. And lastly, let me emphasize that the approach is not limited even to disease-causing pathogens, including not only viruses, but bacteria and what have you, but it is applicable to many other diseases. It can be applied to neurological degenerative disease, such as Alzheimer's. It can be applied to cancer, perhaps one day even to the cause of aging. And so, for that reason, the existence of an approach such as this one raises the important question: will we be able one day to eliminate all human disease? And with that, I thank you for your attention.
Thank you very much, Professor Kornberg. And could you please remain on the stage? We had such a great, remarkable lecture, and I think the answer to your question should be yes, we can end the disease, hopefully, sometimes. So now it's time for some discussions. So if anyone has any questions, this is a great opportunity to get your question answered by the most knowledgeable person in the field. So I would encourage students and faculty members and everyone, if you have questions, there are microphones in front over there. You could rise up and ask your question away. So if you don't know the wording in English, you can ask in Thai language so that we can translate it.
And to the young people in the audience, let me say, you needn't limit your questions to the content of the lecture, but if there are other things you'd like to know about a life in science or anything at all that comes to mind, feel free to ask. And especially like to address questions from young people concerning whatever issues concern you.
ค่ะ เชิญที่ไมโครโฟนด้านบนได้เลยค่ะ อยู่ตรงทางเดินนะคะ ตรงกลางค่ะ
And lastly, might I say, feel free to ask your questions in Thai language. I apologize I can't answer them in the same language.
We can translate the answer as well.
But they will be translated for
So,
me.
any language,
So if
just
you're
get
more
it going.
if you're more comfortable asking a question in Thai, feel absolutely free to do so.
Yes.
Let me introduce myself. My name is Nico from Chulalongkorn University, and it's very nice to meet you here. And I saw on your previous interview with the chief of editor of from Nobel Prize, and I heard that you kind of have a privilege to work with many Nobel laureates in the labs in your previous opportunities. And I also found that your father was a Nobel laureate too. And my question is, may I know what are the common research attitudes that you found among these Nobel laureate scientists? And the second question is, what should we propose as a new graduates from a Southeast Asia countries if we want to work under the supervisor of the Nobel laureate scientists? Thank you very much.
Sure. So the first question, what are the attributes of the people that I have known who won the Nobel Prize, or the very many other great scientists that I have had the privilege of meeting, knowing, or even studying with over the years? And one of the many answers is the freedom to question, including to challenge existing or established ideas, presumably established facts. And I often say that young people should be encouraged to question in this manner. I think the most important thing for them is a kind of healthy disrespect for their elders. This is one of the reasons why I'm always reluctant to speak to young people, but rather to hear from them. I don't want them to necessarily rely too heavily on what I tell them, but rather I'd like to hear what ideas may come to their minds. Healthy disrespect for one's elders. That is a key to the advance of science and discovery, because it turns out so often what we believe to be true is either incorrect or only part of the truth. I would offer as an example what I mentioned today, that the discovery of small interfering RNA. So 20, 30 years ago, we believed that we understood the regulation of gene expression. It was due to what I have described, namely selective reading of the genetic information, and nothing more. Much more recently, we discovered that there is important, indeed crucial, regulation of gene expression at the end of the process, after the RNA is made, when in every cell of our bodies there are small interfering RNAs that destroy RNA, that destroy products of gene expression not needed in that tissue at that time. So we thought we knew everything, and it turned out that an absolutely crucial aspect of the process remained to be found. The other question you asked was about getting to work with and being exposed.
Professor, I think the position that you're standing, it might have some feedback with the speaker. Yeah. Maybe move.
So
okay. And this question is very important. We need to hear the answer. Could you speak up so all of us can hear the answer together?
So the answer to that question is, in a way, also very simple. And it is, there are so many wonderful scientists around the world, including in this university. All that matters in your education is the opportunity to work alongside one of those very many people, and you gain all the benefits that I did from my teachers, and to which you alluded in your question earlier on. Apprenticeship is crucial. Working, it isn't enough to study, it isn't enough to learn. For some reason, actually working alongside a fine scientist is an essential part of education. But again, I emphasize, there are many great scientists. The best scientists that I know did not win a Nobel Prize. The people I most admire in science did not win a Nobel Prize. They are everywhere. And you don't have to look far to find them.
Excuse me, I think there's some problems with the sound. And for the next question, maybe we would like to have someone to ask questions. I see some hands. You can go to the microphone and ask questions. And when answered, Professor, could you move to the position that get closer to the microphone? Yes, yeah. So put the microphones upper, higher up, so that it's closer to the lips. Is that? And then the position might be towards the front. Mr. Uwe suggested that. Yes. So what is the question?
Okay. First of all, thank you for your interesting lecture. I am a graduate student from Kasetsart University. As you mentioned earlier, the siRNA is a great way to get rid of disease. So my question is, can we apply them, like how to manipulate them to improve the anti-aging activity in humans?
So the last part of your question, can we use siRNA
to improve the anti-aging activities in humans?
So, in order to make the best possible siRNA, we survey the entire length of a gene and find the most sensitive points and make the best possible siRNA for that particular therapeutic purpose in that way. So in the case of SARS-CoV-2, for example, many genes of the virus would be candidates for destruction and therapy. We chose the gene for the RNA polymerase of the virus itself. The reason is that it is most essential for virus multiplication, but also because it is most conserved. The siRNA we made recognizes the sequence in that gene of the virus, which is identical in every variant that has arisen. In the original SARS-CoV-2, all the way to the most recent BA series of Omicron variants of the virus. So in that way, the siRNA that we chose for a SARS-CoV-2 therapy has never had to be changed, and will probably never need to be changed, and will be all effective against every future variant of the virus.
Thank you.
Thank you very much. I think we have more time. So the next question shall be up there on the microphone.
Good morning. No, good afternoon. My name is Nat from the Department of Biochemistry. Thank you for the wonderful lecture for us today. And I have quick question for two questions. First of all,
I think please limit yourself to one, just so that one of the other young people gets a chance. Please.
Okay. So, as a siRNA, right? How do we know that the siRNA, when we inject the siRNA of the viral siRNA into our body, it will destroy viral genome, since the virus can be mutated and evolve easily.
So I want to be sure I understand your answer. how do we know if we introduce that siRNA that it does not have another effect? Is that?
Yeah, how, yeah, if it affect pretty well, right? How do we know that it is from our siRNA that introduced to our body?
So, I apologize if I didn't fully understand, but I think you're asking about a question about specificity. Is that right?
Yeah, because of the virus that it is easy to mutate, right?
The microphone makes it difficult to hear. Did I understand correctly that it's a question about the specificity of the siRNA?
No.
Thank you.
Can you help me?
So the question is about the mutation of the virus, right?
Yeah, the mutation of the virus.
Yes.
Yeah.
So, in the first place, when a mutation occurs, we will know because the sequence of the virus, any new strain of the virus will be immediately determined. So if there's any change that would affect the capacity of the siRNA to destroy the viral message, that will be immediately apparent, and the sequence of the siRNA changed accordingly. And remember that, as I mentioned, we make the siRNA by an automatic process. We simply type into the machine the sequence, and the corresponding siRNA is produced. If there is any variation that would affect any mutation in the region that is recognized by the siRNA, we will know that immediately and be able to make the appropriate change. As I mentioned, fortunately, in the case of SARS-CoV-2, the region we chose never undergoes any mutation. It is so essential that any change in that region kills the virus. But the question you ask is nevertheless important, because there will be examples where changes occur, but the important thing is the ease and the rapidity with which we can address that change. Unlike any other therapeutic, we can correct the siRNA overnight to solve the new problem that emerges. Did I answer your question?
Yes, thank you very much.
Thank you. I think we have one more question, and that might wrap up
Yeah,
this
the
evening.
young
I
lady
saw
sitting in
some
front,
hands
please.
and some students. If you have more questions, maybe after this.
Good afternoon, sir. My name is Pisini Bidewong. I am a pre-medical student. Well, I have two questions today. The first one is, are there any side effects of using small interfering RNA? And the other one is, could you suggest us about how to work and succeed like Nobel Laureate? Thank you.
So I can briefly
Can you
answer
hear?
the first
Can you hear the
question,
questions
so I will
well?
allow the second one. The answer is that the Nobel Prize was given in 2006 to Andrew Fire and Craig Mello for the discovery of siRNA, small interfering RNA. Yes, there was a prize, and the only problem was siRNA was discovered by Andrew Fire and Craig Mello 30 years ago, but it is only now, with the approach that I mentioned, that it is generally applicable for therapeutic purposes. And I agreed I would answer your second as the last question, but I didn't understand it. If you could just repeat it for me.
Would you like me to repeat the first question, right?
Say again?
Yes, please.
Are there any side effects of using small interfering RNA?
Well, that was what I said a moment ago. Not for using, the discovery of small interfering RNA was awarded the Nobel Prize in 2006.
Thank you so much.
You're welcome.
We still have some time. Go ahead.
Okay, so me and my friends, we are high school students, and we would like to ask you some questions. The first one is the small interference RNA. From a small Google search, think it's a short-term prevention for viral infection. Like it's in the cell just only for about 5 to 7 days. So I would like to know if there's still need for long-term prevention, or how can siRNA works in the long-term prevention of being infected or other disease too.
So the answer to your question about the duration of effect is very important. So small interfering RNA persists in cells for many days to weeks. In the case of a therapy such as I mentioned for viral infection, it may only need to be introduced once, because within a few days the virus is gone. But if the purpose is protection, then it would be advisable to inhale it once every several weeks, which is of course not at all difficult to do and entirely safe. siRNA can also be introduced in a way which will last for months. I think that the approach that I outlined for you, that would require renewing the effect every few weeks, is probably most desirable.
Yeah, okay, and another question.
So my apologies to the other questioner who I did not allow to ask a second question. Okay. As a student, let me invite you to do so. Go right ahead.
Okay, so my question is that when you're looking at the structure of the RNA polymerase, when you use the X-ray crystallography to look at its structure, how do you confirm that the structure you receive from the data is the full structure, because there are like many subunits? So how can you confirm that the structure you received is the full structure of the RNA polymerase, not just like some subunits of it?
So the object of study was the entire RNA polymerase, including the gene DNA and the RNA. The crystals not only contained the entire multi-protein RNA polymerase and DNA and RNA, they are even the molecule, the assembly, this complex is even active inside the crystal and will continue to make RNA if we allow it to do so. The structure that is solved then is determined by scattering X-rays, which is the same like as scattering light of very short wavelength. Scattering X-ray light off the crystals and recording all of the scattered information. It can be traced back to the source to reconstruct an accurate image of the molecule itself. So that process, whereby the scattered information is used to reconstruct the source of scattering, is called Fourier transformation. It's a mathematical manipulation and can be done today very rapidly and reliably in any case. As it happens, at the time when we did this, it was with the computers available at that time, not at all a rapid process. It took a very special computer many weeks to calculate one structure. Today, it could be done in a few minutes on a laptop computer.
Thank you so much.
I think we have the follow-up questions because we've been waiting for. Yes, you can go ahead.
I have a question about how after we put cholesterol derivative into siRNA ทำเป็นภาษาไทยได้ไหมครับ
ได้ค่ะ
ผมสงสัยว่าเซลล์ใช้กลไกอะไรหลังจากที่เราใส่อนุพันธ์ของคอเลสเตอรอลลงไปใน RNA ครับที่โปรเฟสเซอร์ได้ presentation ไปแล้วสงสัยว่ากลไกนั้นมันแบบ universal แล้วมันสามารถใช้ apply กับอนาคตเช่นแบบยาที่เบสจากโปรตีนอะไรจากโปรตีน จากโปรตีนอะไรแบบนี้ได้ไหมครับสงสัยว่ากลไกที่เซลล์มันเอาRNA เข้าไปหลังจากเราติดคอเลสเตอรอลมันคืออะไรแล้วมันใช้กับโปรตีนหรือใช้กับยาอื่นในอนาคตได้ไหมครับ
I think he's asking about the mechanism of the cell taking in the drug system would be applicable to protein molecules and other type of molecules. Just the mechanism that the cell takes in the MNM. Yeah.
So the question is whether the method devised for introducing siRNA into cells is applicable to other molecules such as DNA and protein. And a very important question, and the answer is yes, and especially for the introduction of proteins, more particularly small proteins called peptides. And the reason why that matters is because it is estimated only 20% of potential drug targets are accessible with the use of small molecules like antibiotics. The remaining 80% are considered undruggable, but they could all be accessed with the use of peptides. So our newfound ability to deliver peptide drugs will transform all of pharmacology. It will become possible for the first time to make drugs to treat, even cure, 80% of diseases that are untreatable today. I might mention that the value goes beyond that. It's not necessary in those cases to discover a drug because we know the sequence of the peptide from the protein, which is the target. And we only need to, again, punch that information into a machine which will automatically make that peptide, and we will have a drug for that target. And in that way, as I say, 80% of all targets in human disease inaccessible today. Another reason why we may one time, someday, remove that question mark from the end of my title of this lecture.
Okay. Thank you very much. I think that's about wrapping up all the questions during this session. And from the question that students are answering, I think that we can kind of feel that the students are really taking in all the information today. So I would like to ask Professor Kornberg to remain on the stage for a little while. And to end today's special lecture, I would like to invite the Vice President of Kasetsart University, Associate Professor Ladawan Pokjit, once more to present the token of our appreciation on behalf of Kasetsart University. On the stage, please.
ค่ะขอเรียนเชิญรองศาสตราจารย์ดร.ลดาวันพวงจิตรรองอธิการบดีมอบของที่ระลึกแก่ Professor Roger Kornberg ค่ะ
Thank you very much. And for this occasion, I would like to invite the Dean of Faculty of Science on the stage to give a closing speech.
ขอเรียนเชิญรองศาสตราจารย์ดร.อภิสิทธิ์สงสะเสนคณบดีคณะวิทยาศาสตร์กล่าวปิดงานค่ะ
Ladies and gentlemen, on behalf of the Faculty of Science, Kasetsart University, I am delighted to deliver this closing remark on the successful special lecture by Nobel Laureate for Chemistry on the topics of the end of the disease, the extraordinary developments in biomedicine and the implication for humanities. To our keynote speaker, I am grateful for his insight shared by Professor Roger D. Kornberg today. His expertise has opened doors to innovative possibilities in biomedicine and the quest to overcome disease. To our valued guests, sponsors, and participants, your presence and engagement have enriched this gathering, inspiring it with a spirit of curiosity and collaboration that defines academic learning and its finest. I do believe that this special lecture can cause further change among participants by using today's knowledge as scientific inspiration for improving the quality of life and all mankind. Finally, thank you all again for being part of this remarkable event. I wish you safe travel and may our shared knowledge continue to shape brighter future for all. Thank you.
Thank you very much. And ladies and